Okay. Thank you, everybody. Greetings to all and best wishes for 2024. So this is the first seminar for the year of 2024 and the 10th of the series Crops to Hunger, Case Studies in Africa and Beyond, supporting CGIAR partners through genotyping services. So thank you for your time and for your presence. So in our previous year, we had 9 seminars tackling crops such as potato, sweet potato, cowpea, sorghum, maize, and others, and how our CGIAR CIMMY genotyping services and support are supporting and being used by our partners. From these seminars, we learn a lot, and also the importance of how reliable genotyping accelerates the decisions on the diverse continents observed. So today we have with us Dr. Manje Godas, Dr. Sudha Nair, and Dr. Suekai Chang from the Global Maize Program, working in Kenya, India, and Mexico, respectively. So our colleagues will be introduced by Professor Mouez Amri from the University Mohammed VI Polytechnic University in Morocco. So Professor Mouez have a PhD in biological sciences and plant breeding, holds a HDR in agriculture science and crop production, with extensive experience in plant breeding and genetics. He has a track record of releasing high-yielding legume varieties in several regions, and these are widely adopted by farmers and endorsed by seed companies. So, Dr. Mouez, thank you very much for accepting our request, and I give you the floor. Thank you.
Thank you very much, Anna. It is a pleasure with me to kick off this 2024 seminar series, especially with one on breeding. I mean, with the 3 of the colleagues working with CIMMY, and especially in one of, as we are based on Africa, so one of the global crop, I mean maize, with an important, as an important food crop for Africa, but also at the global level. And just to make an introduction about this subject, so everyone or everybody knows that maize is an important food crop with a global dimension. So these last decades, there is significant improvement that have been made and achieved with a specific or through specific research and development initiatives, and the development of high new yielding varieties adapted to climate change, resistant to major pests and diseases, and with high nutritional value were really developed and reached farmers' field with a big impact there. However, with all the challenges that we have, such as climate variability now, with the regularity of precipitation, there is more an increase in temperature. We have new pests and diseases still impact the maize production, pushing the ongoing research activities and efforts to invest more in the development of new resilient varieties to improve and improve farmers or farming practices.
Professor, you can share your presentation.
Yeah, I will do. So since CIMMY is leading this global maize research innovation to increase production and productivity through breeding advancement, genetic resources management, and capacity building, we have today 3 eminent scientists that we working on maize breeding or involved in the maize breeding, represent the African continent, the Asian continent, and the Americas. I mean, the Latin America or the American continent. So we have Manje Godwe, Sudha Nair, and Dr. Zhang, that they will be talking today about what they are doing as research in the global maize program. Before we get into genotyping, I would also it would be better to introduce or to more talk about phenotyping before we get into genotyping. And one of the platforms in Africa that could be used to phenotype any of what we have as crops. So in Africa is the Phenoma phenotyping platform, which is high throughput phenotyping. I don't know if you see my screen here. Okay. So this phenotyping platform, which is the Phenoma platform, it's high throughput phenotyping built in Morocco. It is one of the largest phenotyping platform, close to field phenotyping platform, but also with the an indoor component there. Just very quickly, I will go and try to get into the next slides to talk a little bit quickly and try to introduce. It is a lysimetric system with all robotic system where we have these mini plots here, which are somehow representing not a single tube for single plant phenotyping, but it's more mini plots that we can use to phenotype populations of plants with all these robotic systems that could come and take the weighting and adjust the irrigation based on the water regime that you fix. So with this system, we have a first source of data, which is the lysimetric data that we can use to calculate the transpiration rate and all other parameters that we are using. With this platform, we have another one, which is the PhenoMobile machine. Here, with a set of sensors, we have LiDAR, RGB, infrared camera, and spectrometer that we use to collect the upper part, and different images and signals that we can use also to generate and collect a set of data, especially when it comes to the biomass, biovolume, and all other parameters that we can use to assess the impact of any of the water treatments or whatever as a treatment that we have. This is the large screening and close to field phenotyping part, which is the indoor system. We have another outdoor system, which is the PhenoMarzuga. We can use these machines to simulate any future scenario of climate change, especially when it comes to drought. You know that we cannot, when we talk about drought, there is many simulation studies that they mention that in the future we will have more impact of high VPD, which is high vapor pressure deficit, which is a drought in the soil compared to what we have as drought in it's drought in the atmosphere compared to what we have as drought in the soil. So, and then with this machine we can use to apply different stresses or different level of stresses, which is the high VPD and the drought in the soil, combine both together or apply in two different cycle levels. So this is for more digging into the mechanism of resistance and try to look for which trait which trait it's more associated with genes, so we can explore more the mechanism of resistance and dig into the genes there. With this, we have another platform, which is the speed breeding and growth chambers. So this platform, you know that now we are talking about accelerated genetic gain and then how we can accelerate the genetic. So with these few slides, just to show you that we have in Africa this kind of platform that we can use to build the partnership and collaboration, and then it is open for any institutions from Africa or all over the world to get in. Thank you very much. It was very quickly, and then it is time, I think, to move to the presentation from our colleagues here. So I need to stop sharing, and then I will ask. So before we move to the presentation, I would like to introduce our colleagues today who will be presenting an impact on the low and middle density markers genotyping on CIMMY tropical maize breeding. So we have three colleagues here. Dr. Manji Gowda. Manji Gowda holds a PhD on molecular breeding. He joined the Global Maize Program at CIMMY in 2013. Actually, he is based in Kenya and contributes to the implementation of genomic selection and the development of molecular markers for various maize diseases and parasitic weeds in CIMMY Eastern and Southern Africa Maize Breeding Program. We have also Dr. CGIAR Zhang. Dr. Zhang holds a PhD in applied quantitative genetics from the Chinese Academy of Agricultural Science. He is a senior scientist and maize molecular breeder at CIMMY Global Maize Program based in Mexico. He joined CIMMY in 2011, and since 2015, he is leading the maize molecular breeding program or lab in Mexico. where he is implementing modern molecular breeding tools to dissect the genetic architecture of the major maize complex traits associated with abiotic stresses and also the nutritional quality or value. The least and not last, so Dr. Suja Nair, Dr. Nair holds a PhD in genetics from the Indian Agriculture Research Institute, New Delhi. She is a principal scientist and maize molecular breeder at CIMMY Global Maize Program in India, and she is currently leading the maize molecular breeding program for Asia, and also the CIMMY IITA Maize Trait Discovery Pipeline. So let's welcome all the three presenters today, our guests, and then I will give the floor to the first presenter. So please, Dr. Suja, can you share your presentation and stuff? Thank you very much. The floor is yours.
Greetings of the day, everyone. Thank you, Professor Moise Samri, for our kind introduction, and Anna for extending this invite to us to present our work on the impact of use of molecular markers in maize breeding, in CIMMY maize breeding pipelines. So this will be a joint presentation by me, Shwedzai, and Manje. So I'm not able to move this. So I will just stop sharing and share again, okay?
Okay, Sudan.
Please, yeah.
I hope.
Yes.
Yeah, now it's going. Yeah.
Yes.
Yeah. So the brief outline of our presentation will be, I'll start off with our maize molecular breeding organization and our work profile, and the genomic resources and genotyping platforms that we have developed or we are using for our work. And then Shwedzai will take over and he will discuss on the trait marker pipeline discovery and deployment. And Manje will discuss on genomic selection and marker-based quality control, and we will summarize the talk finally in the end. So I will quickly go through how our molecular breeding is organized within our breeding program. The breeding team is led by our director, and we have breeders located in the three regions that we operate in: Latin America, Eastern and Southern Africa, and Asia. In Latin America, we have 4 breeding pipelines led by 2 breeders. In Eastern and Southern Africa, we have 5 breeding pipelines led by 5 breeders, and in Asia, we have 3 breeding pipelines led by 3 breeders. And we have trait specialists, phenotyping specialists, and product development breeders working along with them to develop the impactful varieties that they produce. So we molecular breeders are also, one second. Yeah. So we molecular breeders are also regionally located, and we strategize molecular breeding applications in the respective pipelines in the regions, apart from doing the core research on traits. And we are ably supported externally by our expert biometricians and quantitative geneticists. So this is how we are organized. And this will show, this is a graphic showing a rough outline of our operations of all the breeding programs. All the breeding pipelines. So the stages at which molecular marker intervention is there throughout the pipeline, starting from making of the initial process F1 quality control genetic purity. We do forward breeding for simple traits in early generations, and also in DH lines wherever those traits are available and they are breeding relevant. And some breeding pipelines run closed recurrent selection-based rapid cycling genomic selection populations for improving specific traits for source population improvement. And at early stages of testing, we do genomic selection for line selection for stage advancement. And in new GS strategies that we are following for early recycling also, we will have more discussion on this towards when that topic is discussed. And towards the later stages of hybrid testing, again, QC is rigorously followed for genetic purity and identity of the hybrid parental lines and the hybrids, and we maintain reference profiles for all the hybrids that go out from the breeding pipelines. So this is just to show how the molecular breeding work is embedded within the breeding pipelines, unlike many I have seen where public breeding programs, where that kind of that doesn't happen. So now I'll talk about the genetic resources and genotyping platforms that we have developed or we have been using, which will be of special interest to our partners. So we have been using genotyping by sequencing for most of our high density genotyping needs. Genotyping by sequencing is one of the most widely used reduced representation sequencing methods. And it has been extensively applied for sequencing of more than 17,000 maize materials. And this data is available in public domain. With approximately 1 million imputed SNP calls mapped to the reference genome. So 3 generations of maize haplotype maps are constructed using this data and have been applied to many genetic studies. So CIMMY has assembled 5 association mapping panels, which have been genotyped using GBS and used to map various traits of breeding relevance. And many of our partner institutions have used these association mapping panels along with our high density data for discovery of markers for traits which are specific for their breeding programs. Also, 95% of our released CIMMY maize lines. CIMMY maize lines are elite lines that come out of our program that are international public goods, and they are genotyped using, we have 647 of them as of now, and they are genotyped using GBS. And this data was excellent in clustering the CMLs to 3 structure groups, aligning with their adaptation to lowland, tropical, mid-altitude, subtropical, and highland environments. For mid-density genotyping, we have used 2 platforms. The first one we started using was RAMP-seq, which is a methodology that uses conserved regions for amplifying thousands of middle repetitive regions, followed by scans to identify sequence polymorphisms in low copy intervening sequences. This is a cost-effective mid-density genotyping strategy for large-scale genomic selection projects. And for maize samples genotyped for genomic selection, approximately 7,000 tags were generated on an average, but they are not mapped to the reference genome. So we started routine use of genomic selection as a strategy in maize breeding pipelines in 2017, employing this genotyping platform. But we have moved away from this platform, and currently we are using DART tag platform, which was developed using SNP sequences that were submitted to DART. Around 7,000 SNP sequences were submitted for development of this panel, and we had a version which we also used in the beginning, which was around 2,000 genome-wide SNPs. And currently, the second version has 3,305 genome-wide SNP markers, and they are developed from sequencing data of more than 10,000 breeding lines and landraces belonging to different breeding programs from the 3 regions. And so mainly derived from whole genome resequencing data, GBS data, DART-seq genotyping data, and maize HapMap3. The average marker density of this panel is about 1 SNP per 0.72 MB, distributed across all the 10 chromosomes. And this also comprise trait markers from several discovery studies, along with random markers spread across the genome. And they also include 70 QC markers that we have. So this panel is currently being used for genomic selection, genetic diversity studies, QTL mapping, and developing reference profile. And these are available in public domain in this site within Excellence in Breeding. Apart from this, we have maize low density genotyping SNP sets. So these include trait markers for breeding relevant traits for different product profiles, which are outcomes of trait mapping studies at CIMMY and IITA. And it includes a common set of 50 SNPs used in quality control at various stages that I mentioned before. And also EIB low density genotyping service is a shared CASP genotyping platform serving all CG centers and partner programs. And these trait-specific markers and QC markers are available in the public domain. And these are used for marker-resisted backcrossing or forward breeding, as the case may be, and quality control. So a work that is just completed and not yet in public domain is high quality reference genome, development of high quality reference genome of CML495, which is a key tropical maize line that is assembled with a genome size of 2.21 GB and approximately 37,000 genes. So the quality of the genome is comparable to any published genome with the scaffold length of 221 MB, an LAI index of 24.4, and BUSCO percentage of 96.5. So this actually makes it a high quality tropical reference genome. And in addition, we have also 90 key CMLs where this is a work that was in collaboration with Shanghai Academy of Agriculture Sciences. In addition, 90 key CMLs were resequenced at 10x coverage. And CIMMYT has been working on improving drought stress tolerance in maize for more than 50 years. So based on the resequencing data of these 90 CMLs and their performance to drought stress tolerance, selection signature analysis were performed, and around 600 candidate genes relevant to drought tolerance were identified. So the reference genome, the reference genome assembly of CML495 and the data from these resequenced CMLs will form a valuable data set for developing and improving our genotyping platforms, especially for tropical maize. Now, Shwedzai will talk on discovery and deployment of trait markers in breeding pipelines.
Okay, so I will be the host, right? And
could you please
yes.
Okay.
So
share your presentation?
Thank you.
Okay, it's okay now?
Put in presentation mode, please.
Yeah, please.
Yes, thank you.
It's okay.
Okay, yeah, thanks, Suda, for introducing the genomic resources and also the genotyping platform that we are currently using as the global maize program of CIMMY. And today I will mainly talk about the marker assist selection application. So yeah, for the marker assist selection deployment, first we have to develop the trait associated markers and also deploy the new breeder ready marker for marker selection in maize for forward breeding or marker assisted backcrossing breeding. At CIMMY, in the different geographies, we focus on the improvement of different breeding target traits. So in order to develop the breeder-ready markers consistently, a standard trait marker pipeline was developed by the maize marker breeding team at CIMMY to detect the trait-associated marker for deployment. In general, it has three stages. The first stage is discovery, and then the second stage we call the validation. Then the last stage is the deployment. In stage one, we mainly use the GWAS or the association or the linkage mapping to dissect the genetic architecture of the target trace. If the target trace is controlled by some major alleles with big effect, then we'll decide to do the next step. So in the stage two, then we validate the major QTLs with big effect in the different populations, genetic populations, and also we validate their effect and also their allele frequency in the breeding populations to develop the breeder-ready markers for deployment. So once we have the breeder-ready markers, then we start the deployment stage. Currently, we have several, mainly we have 4 traits under deployment stage. The first is the maize stick wears, and then the maize laser necrosis, and then also provitamin A and the tar spot complex. Then the first one, the MSV and MLN, is a major disease in Africa, and then the provitamin A that was developed for 10 years ago, and then tar spot is the major disease in Mexico. Besides of these 4 traits, and also we developed also other traits for some simple genetic architecture traits like the green colors and then the copper colors and others. In general, our molecular marker deployment strategy depends on the allele frequency and also the effects sets controlling the target traits. So if the target traits controlled by some major alleles with large effect and also their allele frequency are low, and then we prefer to using the markers for marker-assisted backcrossing selection. Then if the traits are controlled by some favorable alleles with moderate to large effect, and then their allele frequency also in our breeding pool are medium to high, and then we would like to apply the forward breeding. So today I will mainly talk about the marker assisted backcrossing and also the forward breeding, and then Mandy will talk about the genomic selection strategy we are discussing. In terms of the MSV, and then Suda in 2015 developed the production markers for the MSV1 on chromosome 1. We combine the GWAS with the linkage mapping. Then we detect major QTLs on chromosome 1. Then after that, we follow a QTL isogenic recombinant strategy to fine map these major QTLs to an interval of 0.87 centimorgan. Then we validate the marker we developed, and then in the breeding lines. Then we confirm that the accuracy is achieved to 94%. Currently, the production markers, 3 production markers, I think is being routinely used in forward breeding in several breeding pipelines. So the second trace I will talk about the MLN, and then this one the allele, actually the favorable allele is from the KS23-6. It's a line from Thailand. So the frequency of this allele detects on the chromosome 6 is not very high. So we cross these lines with 3 CIMMY lines, and then we develop the 3 ditch populations, and then we use a modified QTL-seq experiment. Then we fine mapped the QTLs on chromosome 6. We 2 plants, and then also we developed the markers. From here we can see this is one of our major lines, and then this is the case 23-6 from Thailand. After we developed the breeder ready markers, and then we implemented the marker assistant backcrossing applications for these major QTLs, and then right now we have been introduced to backcrossing these alleles to more than 100 elite lines in which was being used at CIMMY. From here you can see that the major difference, and then this one carried the allele favorable alleles, and then this is the negative of the lines we didn't carry this favorable alleles, and then here we can see the segregation on these favorable alleles. So currently we have converted more than 100 lines in Africa, led by Manji. Also we are working on forward breeding for both MLN and MSV, as I mentioned that. So the MLN before was from the case 23-6, then the allele frequency is not very high in our breeding pools. But after several years improvement, and then the allele frequency is high, so we are able to do the forward breeding on both MLN and MSV. And then in the past 5 to 6 years, and then we are routinely using the forward breeding approach to enrich the allele frequency for both MLN and MSV. In last year, we screening more than 25,000 ditch lines to enrich the favorable alleles. So the last one, I will move to the other major disease named tar spot complex. It's a major disease in the Central and Latin American. So this is the symptom of this disease is caused by 3 major fungi. So in the donor lines of CML 495, and then we used we detect major QTLs on chromosome 8, and then combined with GWAS and the linkage mapping. We found these major QTLs, and then followed by a recombinant derived progeny approach. We found this the QRTS8-1 in a 721 kb interval regions, and then also we developed the 2 production markers, and then these 2 production markers are being routinely used in forward breeding. And also in this region, we identified 2 candidate genes, then the 63511 and then the 73884 was identified. So based on the resequencing data mentioned by the genome assembly data of the CML 495 mentioned by Suda, and also the resequencing data of the KCMLs, and then we did the candidate gene GWAS analysis, and also we check the mutations between the CML 495 and the B73. Yeah, we in total we have around 28 mutation amino acid mutations in these regions, and then 4 are functional. In the genome, in the candidate gene region, yeah, of 1984, 9814, and then so yeah, we validate that this one is a possible candidate genes for the TAR sport complex. So the CML 495 is from heterotic group A. Then yeah, we also want to get some favorable alleles from B group, and then we identify good donors of CML 576. Then in a DG populations, we detect very major cultures on chromosome 10, yeah, which can explain about maybe 24 to 40% of the phenotypic variation. Then after that, we use the fine mapping approach, and then we defined these markers. We found several markers associated with this one, and then also we detect some we develop some markers in the 1.36 megabit interval regions. Yeah, this one is currently ongoing. At the same time, we also validate the marker effects in the breeding lines for deployment. So I think that's all my presentation. So Manji, the floor is yours.
Manji, please.
Thank you. Thank you.
Just a moment, Maje.
Okay.
Okay, you are the host. Thank you.
Okay, thank you.
Just waiting for the Manji to share his presentation. Please, when you ask the question, please mention to whom you ask. You are posting your question, so we can manage better the QA session afterwards. Thank you very much. So Manji, yeah, the floor is yours, please.
Just a pointer. Thank you. Good morning, good afternoon, and good evening. So thank you, Shweta and Sudha, for presenting on the application of molecular markers. In this section, I'm going to talk about how we are using genomic selection. Here, I'm not going to detail on methods. Mainly, I'm going on application, outputs, and products, what we got from genomic selection. So in our maize breeding pipeline, we are applying genomic selection mainly for 3 objectives. One is prediction and selection of untested inbreed lines. So again, predict and select untested hybrids. So and population improvement through rapid cycle recurrent selection. So how are these? So if you go once, first I will start with predict and selection of untested lines. So in maize, the final products are the hybrids. And in Africa, it's a 3-way cross hybrids. And in Asia and Latin America, it's a single cross hybrids. So it's a complex for us to have same approach everywhere. So initial strategy, what we followed was test of predict of strategy, where at the stage 1, we applied genomic selection, like only 50% or 40 to 50% of the lines were selected, test crossed, and evaluated in 2 to 3 locations, and all were genotyped. The remaining 50% were estimated for their breeding values. And while selecting for stage 2, we consider both, which are phenotypic based as well as marker based selection. So lines with the best breeding values of 10 to 20% were selected for stage 2 evaluation. We started this in 2017 in one product profile, and later on we scaled it into all the product profiles. So currently all product profiles are using this genomic selection at early stage. So this saves almost 30 to 45%. But breeders, we have to convince them, is it real, how it will be. So when we did it stage 1, so when we go for stage 2, this is the results. Like around 20% of the stage 1 lines were selected. From those, when we do test cross with the 3 testers. So around 1,042 hybrids were developed. These were evaluated around 5 to 6 locations with a different management, under optimum and drought. This is the results under optimum. So from stage 2, breeders want to select for stage 3. For stage 3, again, they will choose 10 to 20% depending on their pipelines and breeders. So out of this 20%, when we checked how many are really selected only based on the phenotypic data at stage 1. This is the one out of 205 best hybrids, which we wanted to move to stage 3. So 97 were phenotypic based selection, and the which are remaining 108 hybrids are only breeding marker based selection. That means the lines which are selected based on the GBVs are the used for this combination. That means the mean performance of best 20% hybrid which are moving to stage 3, both phenotypic selection as well as marker based selection, they are equally effective. This is for grain yield. So we observed a little bit like early, like half a day early for anthesis date. So similarly, this is under optimum in Eastern and Southern Africa breeding pipeline. And the same thing when we applied and checked it for drought, you can see that the best 20% of the lines which are moving to stage 3, the lines which come from phenotypic based as well as GBV based are equally performing. What this shows that, so if we use a marker based selection, they are equally effective as phenotypic selection. So similarly in South Asia for heat stress breeding pipeline, here you can see this is here the analysis for individual location. The green one is the performance of the hybrids at with only marker based and performance based on the phenotypic based. So this is also clearly shows these are 3 locations. So equally effective, and except in one location we find slightly higher, but overall when we did across locations, so genomic selection based or marker based selection is as good as phenotypic based selection. This shows that this gives a confidence that breeders can scale it into all the breeding pipelines. So that's how we started. So similarly, even in Latin America, also this is an example that under optimum, the performance, this is the performance of the best lines, best hybrids after we selecting only based on phenotype and based on the markers. So they are equally effective. So even this is even the combined across stress breeding the optimum and drought. So this gives us more confidence that we can go for applying genomic selection. That way we can reduce the phenotyping up to 50%. At the same time, as Sudha mentioned earlier, because of genotype, there's a lot of the technology is coming. So new technologies are coming, marker costs are varying with different platforms. So earlier we were using RAM-seq. So next we started to use a DAT-seq. So we have to convince the breeder to show that if you use even different platform, how much you really lose the accuracy or is it will be the same. So this is an example we did for 4 years of data. So you can see grain yield under drought with the DAT-seq. This is the blue, this is the prediction correlations. Whereas the same lines which are used with the RAM-seq, so prediction correlations are equal or similar or is not so significantly different. So in not only in 1 year, in all 4 years, and not only for a complex trait like grain yield, it's also in a simple trait like anthesis date. So this conveys that when we are moving from one platform to another platform, so breeders should not feel that we are changing too much, and a breeder trust is very important, especially if you want to scale up into all breeding pipelines. So that's how then we shifted from RAM-seq, which is around 9,000 marker based platform, to DAT-seq, which is 3,000 marker based. So now we are applying this one DAT-seq markers in all pipelines. So this is a summary of how it started in 2017. This is a test 50 and predict 50. That means here in a pipeline 1, out of 1,500 lines were used, out of that 850 were phenotyped. That means the remaining were only genotyped and predicted for their breeding values. The strategy was the testing population was fully replicated. That means the 50% was completely replicated. That's our training population. We followed the same approach till 2020. From 2020 till 2019. In 2020, as we discussing with the many experts, so then we moved from, instead of fully replicated training population, we went to sparse design. What did add additional advantage? This added additional advantage, the number of plots which we used with the fully replicated is fixed. But with the sparse design, this will help us to add one more location with the same number of plots. That means same cost, we will get one more additional location data. So that's how our training population, the sparse design started. And prediction correlation, when we compared with the fully replicated versus sparse design based training population, the prediction correlations are almost similar. They're not, we're not lost more than 2% of the prediction correlation. So it gives confidence to go on sparse design based training population. So that we followed till 2022, till 21, 22. And from the last year, we again, it's a part of continuous improvement. We moved on from test off, predict off into test all in a sparse design, and using the markers, get the breeding values. So how we done, we'll explain it in the few slides later. So with this, we have a lot of historical data is available. So once historical data is available, so how can we use the historical data? Here you can see this is a 4 years data. The lines, how they are related. In PCA, it looks like they are overlapping, related. When you check this, the parental lines of the DH lines for each year used. That means, for example, in 2017, around 16 parental lines are, 16 lines are used for those DH population. Similarly, around 20 to 30 lines are used in each year to develop a DH lines. So, but there is only 1 or 2 parents are common between the years. That means we have very less common parents, shared parents between the years. So in that case, if you want to predict 2018 data by using 2017, the prediction correlations were 0.34. Whereas when we used predict of test of strategy, the prediction correlations were between 0.6 to 0.8. So this shows that though it's a quite good, but it's like we don't want to take a risk. So what we did, then we checked to use the 2 years data and predict the 3rd year data. Still, the prediction accuracy is slightly increased, but it's not so high. If this worked, this would have helped us to reduce the cycle length, but that's not possible. So when with this approach, because prediction correlations are moderate, not high. So then what we did, if you want to predict 2019 data, take out 10% of the 2019, evaluate in the field. So you combine with the the historical data of last 2 years, then predict. The accuracy suddenly increased is comparable to what we used to get with the test half predictive strategy. So this shows that one way, yeah, the percentage of training population size can be reduced, but we are not able to get advantage of reducing a cycle length. So this is how we can see that when you increase 10% to 30%, the prediction correlations were increased, and after that it's stagnated. So similarly, this is we did it in Eastern and Southern Africa. Similar work also done in Mexico, in Latin America with short sai. So there also had a 3 years data, but here the breeding is a bit more complex than what we used to be here. For example, if you see this figure, this in year 1, there are 291 hybrids. So year 2, 739 hybrids. Year 3 are 498 hybrids. And these 291 lines were derived from, or are from 11 DH population. Its 11 DH populations are developed from 19 parental lines. So it's very diverse. That means each DH population contributing maybe 1 to 10. So this is more diverse than what we used to have the structure in ESA, Eastern and Southern Africa breeding pipeline. So here also the using historical data and predicting the next year data, the prediction correlations were as we observed similar, it's around 0.36 to 0.38. So yeah, here when we use historical data and predict 1 year, surprisingly we observed bit higher. When we cross-check it, it is mainly because the most of the lines evaluated in this 2015, where there is a high relationship between these. That's why it's because most of them are repeated in next year. So similar strategy. So then here we applied a prediction model like a single environment model versus multiple environment model. Here multiple environment means over the years in the same location. Like this is agua-fria location. This is single environment versus across the year multiple environment. So this will also help us to get even higher prediction correlation compared to single environment model. That means use the historical data. It will help us to improve our prediction correlations. So we have historical data. Here, our biometricians always help us to giving that new idea and using it. Here, there are different models they used to prediction correlation. If we come here, this is a training set. That means 3 years data plus 15% of 2020 data to predict remaining 85%. These prediction correlations, what we observed with the these are prediction correlation with the different models. If we focus only on the GBLUP, so these prediction correlations are around 0.5 to 0.6. So the next question is, if I have 10,000 lines as my training population, do I need to handle everything, or is everything is really important? So when we check that one, for example, the same one, if we are predicting 85% of stage 1 lines of 2020 by using historical data plus 15% from 2020, so only 10% from 2017, 8% from 2018, 14% of the total training population 2019. That means less than 20% of the this is contributing. So now the question is how we can do that, separate it for every time. So that's where our biometricians are working on to make it a user-friendly tool to select those effective or informative subset every time whenever we want to use a historical data. So similarly, using the historical data, making a subsets, the work is optimizing for heat stress breeding pipeline for South Asia. So that is what we talked about, selection of untested lines, especially in a three-way cross scenario. So another is rapid cycle genomic selection. Actually, rapid cycle genomic selection in CIMMY, we started way back to 2011, 2012. So initially, we started with the biparental-based rapid cycle genomic selection. So I'm not going to detail into the procedure. So, but based on that, there are several products came out. For example, these are the 5 lines which are developed through rapid cycle genomic selection, are acting as a currently either released or allocated around around 7 hybrids now in Eastern and Southern Africa. So this is, and these lines also released as few CMLs, accepted as few CMLs. So that one we used it in a biparental based rapid cycle genomic selection. But in here its the main objective was to capture maximum favorable alleles for the trait of interest. So as a part of continuous improvement, we started using multi-parent based rapid cycle genomic selection. Here Schwarzai started this work. Here in each heterotic group started with the 15 elite lines are selected and formed like a half a diallele design to make F1, then F2. So then the F2, then that one used test crossed and evaluated under optimum and low N and drought stress. That was used as a training set. The next 2 cycles, the best 5 to 10% were selected and recombined by using this F2 F2 F3 as a training population. So with this, what we observed, you can see that from cycle, we call that when we phenotyped, we call cycle 0. From cycle 0 to cycle 4, we observed that increase in the average grain yield. So overall, we observed the gain by around 100 kg per hectare per year. So similarly, this is under optimum and also observed under higher gain under drought. Similarly, in South Asia, the multi-parent based rapid cycle genomic selection started mainly for drought stress and one breeding pipeline in another breeding pipeline on waterlogging tolerance. So we observed very good gain, and also the lines developed through rapid cycle, this multi-parent based rapid cycle GS are now part of the elite lines using in the new breeding start, as well as parents of some of the announced hybrids. So in Eastern and Southern Africa, we had a lot of historical data. So, and also we moved most of our breeding program from pedigree breeding to double diploid based. Then we didn't have much option to use the rapid cycle genomic selection. So what we came with after meeting with several experts and the quantitative geneticists and all, we came up with a modified rapid cycle genomic selection. What is the difference with the earlier rapid cycle genomic selection and with this is everything is same, multi-parent based, 10 to 20 elite lines in each heterotic group. We make a F1, then F2, then is a cycle 0. Instead of phenotyping them, we have historical data, so used as a training population. But if you want to use the historical data as a training population, we want to know this is related to our training population or not. Unless breeders bring some new genetics for a new trait. So most of the time, since it's recycled, so we can use that. So here you can see that this is the black dots. This is a training population. This is our F1s of the testing population. This shows that they are highly related. There is a relationship. So we can use that, use this historical data as a training population. So we did cycle 1 and cycle 2, combined it. We completed this in a 1.5 year. So currently we submitted this into DH lines. So waiting to see how that stage 1 will perform. This will reduce the breeding cycle time, and also when you go to stage 1, the best lines again will go and part of the training population. So that will help us to improve our historical data. So this is our process. We started only in one breeding pipeline as a proof of concept. We want to see how it performs. So when we checked at cycle 0, there are 2 testing populations compared to the black dot is a training population. They show highly related. At cycle 1, they're still having high relationships. So we believe what we are predicted and selected the best lines are more relevant. So till now, what we did with the GS, we are able to reduce resources and able to get a similar accuracy as it is for phenotypic selection. But our other objective is to reduce the cycle length. How can we do that? So there are several strategies we are discussed and try to implement. The one is with a sparse design. Here in the picture, you can see when we go for test half, predict half, like the blue one, assuming that this is the training population, which is phenotyped and genotyped, whereas the white cells, that means if the line in a 4 location is not evaluated, not phenotyped, it's only genotyped. So these are different versions of a sparse design we tried. When we compared the prediction accuracy, they are yes, not so different, and also this gives us that the other lines also get opportunity to be evaluated. But our objective was to, with the fixed plots, increase number of location. Here, instead of 4 location, the 5 locations. So now we moved from this sparse design initially started into this, where same number of plots, but increase the number of locations so that we can capture more G by E. So this is a sparse design, or we call sparse phenotyping. So that will help us to only save the resources. The next thing is, how can we combine the stage 1 and stage 2? Then it's the thing is the approach we call sparse phenotyping plus sparse test crossing. That means at stage 1, we use 1 tester in each heterotic group. At stage 3, 3 testers. So instead of that, if we use that stage 1, use a 3 test crosses, but instead of test crossing with all the stage 1 lines, use them in a sparse design. So you are using a sparse for test crossing also, sparse for phenotyping also. So this is the approach that will help us to skip one whole year. So that will, before implementing that, we wanted to check. So since we have a lot of historical data, especially stage 2 lines, so we tested. Juan and Pancho did with our available historical data, assuming that if we have stage 2 lines, these all are completely phenotyped, the performance, the prediction correlation is 1. If we use in a sparse phenotyping, the all 3 testers are crossed, the prediction correlations are around 0.96. And if I use sparse phenotyping plus sparse testing, that means there are one tester is test crossed with all the lines, whereas 2 testers are sparsed. So this will reduce 30% of the total number of plots what we have planned, but the prediction correlations are same. So similarly, when we checked these best lines, if we select based on the sparse phenotyping plus sparse test crossing, they are, for example, if it is completely phenotyped, the mean performance of the best performing 20% lines is around 8.4 tons. Whereas if the sparse phenotyping, it was 8.36 ton, and its sparse phenotyping and sparse testing combined, this is also around 0.35 or 0.37 tons. So it's not much different, and same lines were identified in the best 20% of the lines. So now we are planning to use this sparse phenotyping and test crossing approach with combining with the historical data. So in a summarize, we started the genomic selection with a completely replicated training population in a test of predictive strategy. So later on, we moved that one into same test of predictive strategy, but sparse design is based on the training population is based on the sparse design. So now from 2023, we are testing all in a sparse design. With the sparse design, compared to the completely replicated one, it reduced up at least 30% of the plots. So that much phenotyping cost will be reduced. So we have this modified strategy I explained earlier. We are trying to combine sparse phenotyping and sparse test crossing strategy. So whenever we are coming with the new idea after discussing with these quantitative geneticists and biometricians, we are always starting with one product profile to implement. Once we get a promising result, then we are scaling into all other product profiles. So that is for genomic selection. In parallel, we are also doing QC. We developed the technical manual for quality control, where it involves all the information and markers used. So QC is the mainstay in many stages in all maize breeding pipelines, starting from parental and F1 QC to line finishing. Currently, all the inbreed lines, especially the elite lines, in each regional breeding hubs have a reference profile of these QC markers. And this is also always available for internal use as well as for partners also. Whenever they are using a CIMMY material, they can compare this profile for their genetic purity. And all the CMLs, around 630 CMLs are there, also genotyped with the QC set. This reference profile is also available for internal and partners use. So in summary, the cost-effective genotyping platforms are key to use a novel tools and strategies in a breeding pipeline. For us, especially when we use forward breeding, that low cost, 10 marker SNPs based forward breeding, the turnaround time of 2 weeks is so useful. It helped a lot to cut down the lines which carrying unfavorable alleles in time. So then this application of markers are mainly depends on the trait architecture and whether trait as a must-have trait or not in a selected pipeline, and cost comparing to phenotyping. If it is phenotyping is so cheap, then there is not much use of using the markers there. So maize, we have a stage gate advancement process for any discovery, before we are start to choose any trait, it has to pass through stage gate advancement, where it has to be discussed whether that as doing that any trait, new trait, it is useful or not, it has advantage or not. So based on that, if it is advantage, then it has to pass through each step of the stage gate advancement. So GS is a routine strategy followed in all breeding pipelines in CIMMY, and continuous improvement of the existing strategies are explored for cost and time efficiency with the support from several different teams and also breeders and quantitative geneticists, biometricians. So yeah, it's like we are scaled up, but still we are open to learn and implement new ideas which can help us to improve selection efficiency, as well as it will save the resources and reduce the cycle time. So yeah, we have to thank all the donors and the breeding platforms and excellent EIB and service providers for this wonderful work. So thank you all.
Thank you very much, Manji, and all the team.
So now we can open, we are a little bit behind the schedule, but it's not a problem. So we can start to open for the questions. You can unmute yourself. And after Dr. Prasanna, I would like to invite you after the questions to give some words if you don't mind. So please, Bodo, I think you are the first one to make the question. Can you unmute yourself?
Thank you, Anna. Good afternoon or good morning. I have actually two questions to Manji, I believe.
Yeah,
mostly,
please.
but the others are also asked. First, how many phenotype data points did you have to generate to come up with well-predictive markers? You know, by correlating these things, associating and working it out from your what you call historical data, probably that was collected over the years. This is one question, and the other question is, and I ask this for specifically for a few of my crops, particularly the clonal crops. Do you think that it can be possible to, let's say, predict by genomic selection the next generation of parentals, and then also the performance of these hybrids, and then based on this predicted hybrid values, select these predicted parentals and do a next cycle of genomic selection, all in silico, and then get a prediction for the next courses to develop this parental population without needing to do test courses and making test hybrids. So the two things: first, how many data phenotype, and second, predictive, prediction power. Thank you.
Yeah, regarding how many data, yeah, well, actually, we are using currently, like for optimum, we are using the 2 locations. So that means with 2 replication when we started, it's like a 4 data points, at least minimum we used to have that. So when we apply sparse design, we will make sure that that is that many plots are maintained. Second question regarding, can we use okay, and regarding on the first question, when we are using only 1 location data, especially when you do for stress, which is more expensive, like drought or low soil N, when it's a 1 location, yeah, it's difficult to have that G by E and all. So that's where we don't use the sparse designs there. So that's what I hope I answered that first question.
Yeah, it is also about the need to, because I understand you have to have phenotypes to be able to pick these marker alleles, or let's say the marker sequences that you have that you use for genomic selection then in the next generation within a, let's say, within a closed population of parentals. And then is it possible after some time to drop the phenotyping completely? I mean, I'm overemphasizing now.
Yeah, we can't complete. Yeah, that's what we initially we thought that same way. Like if we have so many years historical data, we can completely replace the phenotypic data and predict it, even if it is some relatedness also. No, we can't replace it. We need to have, because that relatedness is what it matters. So yeah, we can't replace that completely phenotypic data. Phenotypic data has to be complementary.
Okay, and are you continuing to accumulate phenotype data in every generation?
Yeah.
Okay, thank you. Yeah, I mean, this must be said at some point, because some people, I came across some people who have been believing that after some time they can completely skip the rest of their life.
No, we are accumulating. Yeah, even when we shipped from one platform to another platform, so we made sure that the historical data was genotyped with the same platform which we start now using that tag also, so that we should not lose our historical data.
Yeah,
Thank
I'll
you.
comment. I think it's depending on the diversity of the breeding materials in your program, and then if you implement a genomic selection in a closed Gen Plus environment, then yeah, I think yeah, you need less phenotypic data. But if you introduce new Gen plasm into your breeding pool, then you need to get the new phenotyping data, and then to get the relationship between your historical data and the new breeding data, and then to improve your prediction. I think that's my comments. Thank you.
Yeah, thank you. Very good. And now this other question about the possibility to skip the test crossing.
Yeah, we are trying that. Our historical data shows that it's possible to skip. So now we are planning with the one product profile, and also there is a idea. It's like, we started that one. Then there is another idea of combining stage 1 and stage 4 and increase number of locations. But currently, yeah, we are trying to combine the sparse phenotyping and sparse test crossing. We are also eager to know how much how effective is that? Yeah.
Yeah, my comment is for implementing genomic selection, how to optimize your breeding pipeline to integrate the genomic selection into your breeding pipeline. I think each crop and then each breeding program should have their own a little optimized approach.
So there is one question from Godfrey Shigeza. I'm not sure if Godfrey is still here, and Dr. Prasanna already replied, but Godfrey, you can unmute yourself and Dr. Prasanna can reply to you, please. Okay, so the question of Godfrey is that how do you involve the NARS in the GS approach? So Dr. Prasanna, maybe you can...
I think I answered that question. Basically, it is indeed possible to involve national programs as a part of the GS strategy, or national programs to implement an optimized GS strategy vis-a-vis what CIMMY is doing in maize. However, as we all know, genetic relatedness among the materials between national programs and CG breeding programs are absolutely important to understand. This is not something that we can randomly implement. First, in a crop like maize, strict discipline in terms of maintaining heterotic groups is a fundamental requirement. And then having a strong understanding of what are your basic lines or founder lines that you are using for recycling and how related are they? How can we use the training data that has been generated in different programs effectively to predict the performance of materials that are in the national programs? This all revolves around relationships among the germplasm. The stronger the relationship, the greater will be the predictive value. And the weaker the relationship, you may not be able to extrapolate it at all. So that is fundamental. So some programs can indeed optimize this and use the constant information that is generated through CIMMY, and some may not be able to use it at all. It all depends upon alignment on these specific aspects.
Thank you.
Yeah,
Professor
and also,
Luis, you have your hand raised. Do you want to make a question? Or
I would like first to thank all the presenters for the nice work. I have comment or question, you can consider whatever it is. Still, with all the achievements that we have, especially in Asia and other continents, still in Africa, the yield potential is still very low. And I think it goes beyond what we are doing in terms of breeding, especially with the genetic gain or the plateau of genetic gain that for maize that we have. So when you compare the potential yield, I mean, in Latin America or in Europe or in US or Canada, to compare with the African continent, it's big gap. And one of the problems maybe
let me quickly correct on that. Let's not mix up the genetic gain with the productivity at the national level. These are different aspects. Genetic gain, in terms of genetic gain in Africa's stress-prone environments, there is an excellent, there are several studies that we published which shows excellent genetic gain, even under very challenging environments. So constantly hitting 1.5 to even 2% in different breeding pipelines. Productivity at the national level for maize is influenced by many factors, including the economic management practices, the rapid varietal turnover, and the continued prevalence of old and obsolete hybrids in certain market segments. So there are lots of factors related to the low yields that of smallholder farmers face in Africa. But despite that, one important thing is that I would like to highlight. CIMMY's climate resilient maize has grown from almost 500,000 hectares in 2010 to now 7.5 million hectares or more within a span of 13 years. 7.5 million hectares in sub-Saharan Africa out of 42 million hectares. It means that there is still a lot of potential to expand with improved genetics, a lot of work to do, but tremendous strides made by several African countries.
I didn't say the opposite of that. So, but I said still there is a lot of things to be done in Africa, specifically taking into consideration the specific conditions or environment in Africa. Still the gap is there, a lot of things to do, and then a lot of effort. And just if you because I didn't finish my comment or question. The African conditions, there is problem of striga, and this is big problem when it comes to one of major problems that limit maybe the expansion and the yield potential in Africa. I know that there is no complete resistance, as I'm working on parasitic weeds too. So what you did so far with this, especially when it comes to the genomic selection, this is my question. The first one was more comment to introduce my question. So you can respond to my question.
Okay, Melaku, I don't know whether he's still online, but Melaku has been working on striga resistance markers. Actually speaking, the striga resistant lines and hybrids developed at IITA are excellent in terms of yield and capacity to resist the Striga pressure. This has been demonstrated very well. In West Africa, and we in progress with IATA Striga resistant germplasm in our materials, and we got excellent results too. Just last year, there have been extensive on-farm testing done in Western Kenya under farmers' conditions, Striga resistant hybrids versus susceptible hybrids. The differences in terms of phenotypes are fantastic to see. So there is indeed a lot of excellent work done both at IATA and CIMMY. As regards markers, a lot of work is done by Malaku together with some institutions in UK. And Malaku is now validating some of those markers for Striga resistance. But Manje's work parallelly in Eastern Africa also shows that there is an enormous opportunity to do genomic prediction. We don't have to necessarily only go through marker-assisted forward breeding approach. So therefore, on both fronts, there have been some excellent leads. You are very right. Striga is a major crippling factor in some of the smallholder fields, but it is in pockets of Kenya, pockets in Uganda, Tanzania, and in West Africa. It's not uniformly dispersed everywhere, but it's certainly a major issue in certain pockets.
Thank you. I don't know if anybody have any question. Otherwise, we are already 23 minutes after the schedule, but I think it was excellent presentations, also excellent discussions. So if nobody have any question, we came to the end of one more seminar of this series. Thank you very much for your patience. I'm really sorry for the last time in the incident, but we are resolving that. So today the seminar went all well. Thank you very much. And for the next seminar, I hope to find you also here. And we will focus on common beans presented by Eunice Kemis from Nakrinaro. And one more time, see you next time. And thank you to all and mainly to the presenters. Thank you very much.
Thank you. Thanks to all the presenters from the maize program. Very proud of the tremendous work that you do. And this is quite inspiring, I'm sure, to many colleagues across the spectrum. Thank you, guys.
Thank you.